All right. Hello, everyone, and thank you for joining me for this month's Aroma Explore the Wheel. Today, we are going to be talking about citrus fruit, but more specifically than that, we're actually going to be talking quite a bit about bitterness, which, if you know me, you know that bitterness is one of the things that I really love to talk about. So I was going to find a way to shoehorn it into this webinar series one way or another, but it is relevant, I promise. So I know we have a couple of new people here this month. Welcome. Thank you for joining us. And we're going to start. I'm going to, first I have to get this window out of my way so I can get this window in the correct spot. There's a lot of juggling with these. And great news, I have my wonderful husband, Tom, is running my StreamYard for me tonight. So if I accidentally mute myself for, I don't know, the duration of the webinar, I have somebody who can actually tell me that that's going on. Not that you all didn't try. I really appreciate everyone who tried to let me know that I had no audio, but I always have everything on Do Not Disturb. So it's very hard to reach me. So anyway, this webinar tonight, like I mentioned, we're going to talk about citrus a little bit. Mostly we're going to talk about bitterness. So let's get started. First with the code of conduct, you all should have the link from the email that you received today that would take you to kind of our FAQ page for the Aroma Explore the Wheel. You can read the full code of conduct there. Basically, don't be mean to me, don't be mean to each other. Don't steal my work is totally fine. If you like to post about it on social media or whatever, that's not stealing work, right? So I always appreciate when you all do that, and thank you to the people who have. Other things about the code of conduct you'll see in the, or I guess I should say, if you see anyone in the comment section uh, being abusive or in any way violating this code of conduct, you can just pop into the chat, let me know, and we will take care of it. Although this is a super cool group of people, and I don't see that that's probably going to be an issue, but if it is, just know that there is a code of conduct, and if you don't feel comfortable addressing it in the moment, it is always okay to send me an email afterwards to let me know what happened. But like I mentioned, you can review the full code of conduct on my website. So let's do our session overview. This one is going to be similar to the others. So if you've been here before, this is basically going to be the same format that we always use. First, we're going to do our grounding or warm-up kind of routine exercise. Then we're going to go into our first blind smelling exercise. For that one, you will need your vials that you received in the mail. And I did have somebody let me know, again, I sometimes forget that not everybody can read my mind, that your vials, when you open them, they'll have a little like plug on the top. You'll need to unplug that when we get ready to start doing the blind smelling. It's going to be really difficult to be able to smell our aromas with that on. So just so you know, you can go ahead, if you don't already have those ready, go ahead and grab those to get ready. After our blind smelling exercise, we'll go into our flavor discussion. Then we will do a second blind smelling exercise. It's going to be the exact same flavors or aromas, I guess, that you have in front of you. We're just going to, get more exposure for that. We'll do the flavor reveal. I have Q&A here at the end, but anytime you have a question throughout the webinar, feel free to put it in the chat box on YouTube. I will see that and I will be able to answer it. I do not mind at all answering questions in the moment as you have them. You don't necessarily need to wait until the very end to ask your questions, particularly because I know for me, like I'll always kind of forget what I was going to ask. So that's going to be our session overview. And for those of you who are new, this warm-up exercise we're going to do next is something that we do at the beginning of each webinar. It's your pre-evaluation routine. Those of you who have been here before, you're already familiar with all of this, but the point of us doing this routine, taking this time at the beginning, is to calm your nerves, focus on what you're doing, set your intention for what we're doing tonight, which is going to be evaluating blind aromas to determine what kind of citrus aroma you're smelling. And this is really important for a lot of reasons. I won't belabor it too much because I talk about it all the time each month, but with this routine, it's something that I want you to create for yourself. So we're creating this space within the webinar for you to decide what you want your pre-evaluation routine to look like, for you to practice it. If you want to try out some different things, go ahead. This is your time and space to do that. And this is important because I want you to develop a routine that you're going to do at the beginning before you evaluate anything. So if you're sitting down to an exam, or if you're just studying and you're maybe doing like study tasting, this obviously even works for things like written exams, or anytime that you're nervous. If you have a routine like this, it's going to help calm you down. It's going to help you focus. So a few notes about the pre-evaluation routine. Like I mentioned, we want to perform it at the beginning of each evaluation session. And I already talked about these three things. So routine suggestions, and I do this each month. I will show you all kind of what I do in terms of my pre-evaluation routine, keeping in mind that the goal of developing a pre-evaluation routine is for you to figure out what works for you. You don't have to do it exactly the way I do it. There are a ton of different ways to, ground yourself, center yourself. But I'll give you some examples of what I typically do. First, with our routine, I feel, you want to do something, there's a physicality to it. You want to release some tension. For me, that looks like kind of, shaking out my shoulders, stretching out my neck. Like I've mentioned before, my shoulders are generally like right up here by my ears, and my jaw is always clenched. So for me, I like to just make sure that I'm releasing some tension. I also like to do a little bit of a lat stretch. Again, whatever works for you. But I do encourage you to kind of move around to release some of that tension, because even if you're feeling, let's say, very calm, your nervous system may not be, right? So you could be going into something super confident, but maybe you still have some jitters, and actually releasing that tension from your body is going to help focus and help, tell your brain like it's okay, we're safe, we don't need to be tense. I also recommend some kind of a breathing exercise. The ones we've discussed before on here are box breathing, which is when you inhale for a count of 4, you hold your breath for a count of 6, you exhale for a count of 4, and then you hold it at the bottom for just a second or so, and then you get started again. The other breathing technique we've discussed before is alternate nostril breathing. With that one, you're just going to take one finger, close one of your nostrils, inhale through your nostril, exhale through the same nostril, switch nostrils. So you're always switching nostrils on the exhale. This is used a lot in yoga. This is also just a really good grounding technique with your nervous system. Your nostrils actually kind of have different functions. So breathing into your left nostril is going to calm down your parasympathetic system, or activate your parasympathetic system, which is going to calm you down. Breathing through your right nostril is going to activate your sympathetic nervous system, is going to make you more alert, is going to make you more focused. So those are just a couple of breathing exercises. There's a ton out there that you can look up. Those are two of my favorites. Those are the two that have been most effective for me. The last thing I want you to do is set your intention, visualize what it is that we're going to be doing here. So like I mentioned at the very beginning of this, an example of an intention, kind of a visualization, is I am about to mindfully evaluate blind samples to figure out, or to try to identify, what citrus aroma I'm smelling. You want to be specific. For me, it also helps, particularly in exam situations, to be very clear and specific about what you're doing, because otherwise, you don't want to come in and be like, I'm taking the master cicerone exam. Like you need to break it up into chunks, right? So having that intention, being as specific as possible, is going to help you, kind of calm down all the noise, but also narrow in on what is it that is in front of me right now that I'm doing. So what we're going to do now, I'm going to give you about a minute or so to go through your pre-evaluation routine, and then we're going to go into our first blind smelling exercise. So take a minute or so and do your routine, and I'll be right here doing my routine. Okay, I'll give you a few more seconds to kind of come back, get ready for a blind tasting or blind smelling. Okay, so with our blind smelling instructions, this is going to be the first one. And quickly, for those of you who are new here, the reason I just kind of throw you right in is because this is a form of dynamic learning. Dynamic learning is a lot more effective in improving your performance because what you're doing is you're kind of learning on the fly, right? You're messing up and you're learning from those mistakes. So each time you can learn something from that mistake and come back, your experience is going to grow, right? That's also how you build expertise. That's compared to something like static learning, which is like going in and taking an exam. It's going to be a snapshot of what you know in that moment. So I like to throw everybody in. You all are lucky because I've also started giving you lists of potential aromas that you might be smelling. The very first group just had to go in completely blind. I said it's a fruit aroma, so figure it out. And they did a really good job. So now you all at least have lists. So you've got the bit.ly link on there. You also have the QR code. Enter the date. Your 3-digit personal ID is what was written on the back of the card that was in your package. For each vial, there is a 3-digit number on the bottom. Just type in whatever that number is and smell it. Enter the aroma you detect. When you're finished with all of them, click submit. As a reminder, our gaining sensory expertise is not a linear process. We're looking for progress. We're not looking for perfection, right? You're not going to be tested on this. I'm not going to put anybody on the spot. This is for you, but I do want you to be uncomfortable going into this because that's going to be what kind of pushes you in the direction of building your expertise. So as always, I'm going to give you 5 minutes. You're going to take those 5 minutes to smell through your vials, try to identify the aromas. Like I said, I put some citrus fruits on here. I will say that the citrus aromas you have in front of you are on this list. And that's all I will say about it. So I'm going to mute myself, hopefully not for the entire time, and I will be back in 5 minutes. Okay, we're about halfway through, so you have 2.5 minutes left. You have about 30 seconds left. All right, so that was 5 minutes. And go ahead and finish up your forms if you haven't already. Make sure you hit submit. If you can, just go ahead and leave that up because we'll be using it again towards the end. If you've been here before, you know that I do not tell you what the flavors are that you're smelling until the very end. So let's move on to our flavor discussion. For today's discussion, we're going to be discussing bitterness, what it is, how we perceive it, and how it arises in beer. And I think you can probably guess how bitterness relates to citrus fruit. And I wanted to talk about bitterness today because in the previous webinars, and I understand not everyone has been to those, but we do cover hops, we cover thiols, and I want to make sure that I'm giving you a good look at kind of a holistic look at the world of beer and how you can become a better beer evaluator. So many of us have learned that the pith of citrus, that thin white layer that's between the pulp and the peel, is bitter. However, and this is very interesting because I did not know this actually until I started doing research for this webinar. The citrus pith is actually completely flavorless, and the citrus peel is only subtly bitter. So where does the bitterness that we associate with citrus pith comes from? As it turns out, when the pith and peel are abraded together, so let's say you're cutting a piece of citrus fruit or you're zesting it, when the pith and peel get abraded together, acids and enzymes convert these flavorless compounds that are in both into new compounds that have pronounced bitterness, which I think is a pretty cool thing. And that was actually, I went into this like, oh yeah, we'll talk about bitterness because the pith of citrus is bitter. And then it was like, what? It's not at all. It's actually what happens when you kind of disturb the citrus fruit. So that's just a little, I felt like I needed to make some connection for you all between citrus and why we're talking about bitterness today. So we're going to start by talking about our sense of taste. And our sense of taste is one of our 5 basic senses. The other four basic senses are vision, hearing, touch, and smell. Our sense of taste helps us decide whether we should put something in our mouth, or not, I'm sorry, whether once we have something in our mouth, if we should actually ingest it, if we should actually swallow it. So in that way, your sense of taste is kind of your last line of defense before you potentially poison yourself or eat something that's going to be harmful for you. And evolutionarily, that's what they believed our sense of taste was for. We'll talk a little bit. We'll talk in a little bit about how that may not be entirely true. But let's talk about our senses. So, talking about our sense of taste, there are five basic tastes, and they're proposed evolutionary purposes. And I say proposed just because this is, science is changing all the time. We'll talk in a little bit kind of about how our sense of taste, what we thought was only a sense of taste, is actually much broader than that. So we have these purposes that have been put forth that, right now, based on everything we know, they make a lot of sense. So the first one is sweetness. And sweetness tells us about the sugars in something. Sugars give us fast energy and carbohydrates. From an evolutionary standpoint, we need fast energy from sugars for our fight or flight response. That's now more commonly called fight, flight, freeze, or fawn. And of course, calories prevent us from becoming malnourished and starving and susceptible to diseases. So that's what our sense of sweet has evolved to tell us about, that there's available energy and nutrition. Then we have our sense of sour or acidic. It's really interesting about, and I apologize because you'll hear me say a lot that it's really interesting, but like I mentioned, bitterness is one of my favorite things to talk about, and just our sense of taste in general. But when it comes to our sense of sour or our ability to pick up acidity, science isn't actually clear why we have a sour ability. And again, kind of the best guess that we've come down to is that sour can tell us something about caloric availability in vitamins, especially vitamin C. But it can also tell us things like if a fruit is unripe. So a fruit that's unripe is going to be very acidic. We're not going to want to eat it. It can also tell us things like the milk has gone bad because it turns sour, and it can alert us not only to caloric availability and vitamins, but also like, hey, maybe don't eat this. And then we have salty. Salty is, here I go again, it's another interesting one because we actually have two different receptors for salty. For all of our other senses, we only have one kind of receptor. For sodium, we have one for regular levels of saltiness. So just, you're eating a bag of chips or something, and that's your regular receptor, regular saltiness. And we also have a receptor for when something has too much salt. And that is a completely different receptor. So our sense of salty is, basically is foundational for, I shouldn't say salty, so sodium is foundational for our existence, right? So we need a certain amount of sodium. Sodium plays a critical role in balancing our water levels, and too much sodium at one time can be dangerous. So that's why we have this second too salty receptor to tell us like this is extremely salty, this is going to be dangerous. So salty, we have two different ones. So I would compare that, like eating a potato chip to like shaking out salt on your hand and just licking it. You're probably going to activate that second sensor that you're ingesting something that's potentially dangerous for you and too salty. Umami tells us about protein and also assists with neurotransmissions. And umami is our most recently discovered taste sense. And it was discovered in 1908 by a Japanese scientist. It took almost 100 years for umami to be officially accepted as one of our senses. We'll talk in a minute about why that is. But umami detects glutamate. Glutamate is an amino acid, and umami is a signal for protein, which makes sense since amino acids are protein, and it's a fundamental part of nutrition. So for each one of these senses, we've talked about, or there's been research into like, why is it there? And you'll see on this picture I have here, I'm not that old, but I remember this picture being in my like 7th grade science book. I remember doing an experiment in science class where we were, like swabbing our tongues with different solutions. And this tongue map is not correct. It's not entirely incorrect, but it's more incorrect than it is correct. So we don't actually just taste bitter on the back of our tongue or, sweet towards the tip. We actually can taste it all over our palate. And before I go too far into that, I need to finish talking about bitter. So we'll be talking about bitterness in depth today, but briefly, strong bitterness is a marker oftentimes for toxins. So something that's bitter is going to tell us, hey, this is potentially harmful. And that's also why our sense of bitterness, if we do have something that's potentially poisonous, that's why if you take something really bitter, you stick your tongue out, because that you're sticking your tongue out is an actual like evolutionary reaction to something that might be potentially poisonous. It's trying to get it out of your body. So tastes are caused by only a few molecules. And I think that's super cool because just comparing our sense of smell to our sense of taste, taste is basically hardwired into us. Like when you're born, you can put bitter on a baby's tongue. They'll make the face, they'll stick out their tongue. You put something sour on their tongue, they'll have the same reaction. So we really are born with this sense of taste kind of already there. Whereas our sense of smell is developed mostly by experience and memory. So let's talk about this tongue map. As I mentioned on the last slide, that is not necessarily incorrect diagram, but it came from a mistranslation back in the 1900s. And I think it's one of those things that, that thought of we have certain receptors on a certain part of our tongues. At the time, it made sense, right? So it got adopted and it didn't really get questioned. Plus, it's not like we had like a ton of communication. We certainly didn't have instantaneous communication or access to things like we have now. So there wasn't really anybody fact checking that like, hey, does this guy actually know German when he translated it? So with the tongue map, there are 5 criteria that classes of compounds have to meet to be considered a unique basic taste. In some of the literature out there, you'll see that fat is potentially going to be our 7th taste. Metallic, calcium, those are a few other that have been floated. Some of those, like metallic and like capsaicin, the spiciness in chilies, that's actually your trigeminal nerve. So that's a completely different system than what's going on your tongue with your taste buds. So what does it take for, let's say, we want metallic to be a compound? So the first step is that the compounds should have a distinct class of effective stimuli. This means that the compounds consistently trigger a particular response. For taste, the particular response is activation of neural pathways. The neural pathways transmit the sensory input to the brain. So that's our first requirement. The second requirement is that there should be a transduction mechanism that includes receptors to change the chemical code of the stimulus to an electrical signal. So transduction is the process of converting one form of energy into another. By converting the chemical energy to an electric signal, the brain can interpret it as a sensation. The third step, there must be neurotransmission of that electric signal to processing regions in the brain. So neurotransmission is the process by which our nerve cells communicate with each other and with other cells. And neurotransmitters are chemical messengers that bind to the receptors on the receiving cell. and that's going to trigger a response. Next, the compounds should be perceptually independent from other taste qualities. This means the qualities are distinct and can be identified individually. Lastly, there must be physiological effects after activation of taste bud cells. Physiological effects can include releasing digestive enzymes, releasing insulin, increasing your heart rate, starting to stimulate your signal the stimulation of food movement through the digestive tract, and increased salivation. So all of those things are considered a physiological effect after tasting something. And our taste receptors, as you can see here, they're not only in specific areas, they're actually all over our tongues, which is why it's important when you're tasting something, you're evaluating something, you have enough of the sample in your mouth that you can move it around your palate and coat your entire palate. Our taste receptors are bundled up into 3 kinds of papilla on our tongue, but we also have taste receptors on our soft palate. So that's like the roof of your mouth, kind of towards the back of your throat, your epiglottis, and your esophagus. And we're going to learn in just a minute, that's not the only place we have some of these receptors. So our tongues actually have 4 kinds of papilla although only 3 of them contain taste receptors. So our 4 types of papilla are fungiform, and these I think makes sense. So, wrong one. So our taste receptors, the first one is, pardon me, our papilla. So I've got it now, everybody. Fungiform. So that's going to be a form of papilla that we have on our tongue. They are fungiform. They look like mushrooms. So fungiform, they look like mushrooms. Those contain 2 to 4 taste buds. We also have our foliate papilla. Those are grooves that are located on the sides of the tongue. Those contain about 100 taste buds each. And again, I remember foliate because of like foliage, right? It's around the edges. Then we have circumvallate, and these are on the circumference, right, the perimeter of the tongue. They're kind of arranged like an inverted V, and these contain several hundred taste buds. So the circumvallate have a lot of taste buds. Again, I think that makes sense when we're talking about the purpose of our sense of taste is to help us get nutrition and to help us avoid poison. So to me, I was like, yeah, that makes sense that like the ones with the most taste buds are going to be kind of in the back, right? Like the very last chance you're going to have to make sure you don't ingest something poisonous. Then we also have filiform, and those I like to think of, they fill in the space, filiform. They cover the entire top of the tongue. They don't have taste buds, but they do have a tactile, like mouthfeel function. So for our trigeminal sense, those filiform are going to be where we're going to get that trigeminal sense from. So our taste receptors find and grab a particular type of molecule. And if you were in last month's webinar, you remember we talked about some esterases and how they live in like the membranes of the cell, and they'll kind of reach out and grab what they need as it comes by. So the taste receptors are kind of the same. One common descriptor for them is like a lock and key, right? So you can have a lock, it's just going to fit one kind of key, and that's how our taste receptors operate. So let's talk about extraoral taste receptors. One cool thing about our body is that our receptors for sweet and bitter, and actually umami as well, aren't only located in our mouths. We actually have these receptors throughout our bodies. So you can see here this body map of the guy is showing us where we have all of these different receptors in our body. And so taste receptors were originally thought to be located on the tongue only, right? And again, we're thinking back like through time and, trying to explain things particular about our bodies. The taste receptors, obviously, is right there. It's on your tongue. You're eating every day. So you're thinking this is a taste receptors. Of course, as we've learned more through science, we actually have these receptors throughout our bodies. So the term extraoral isn't really correct, because what we're discovering now is that our sense of taste is only part of the responsibilities of these receptors in our bodies. And that's why, again, like the most obvious thing is going to be what's right in front of you, what you can taste. So really our sense of taste is bigger than just what we have in our mouth. And so sweet and umami have the same kind of receptors, and bitter has a different or another type of receptor, but all of these receptors are known as G protein coupled receptors, or GPCRs. These are a large family of receptor proteins that are embedded in cell membranes. So just, the same thing. It's in the membrane. They're going to kind of reach out like catcher's mitt and catch these, what it is that they're looking for. So for example, something that's sweet, right? So our sweet taste, GPCRs, bind to sugars like glucose or maltose. And, the same thing with our bitter GPCRs, those are going to bind to different bitter compounds. So during this interaction, the GPCR initiates a signal transduction pathway, and we just talked about that. And this pathway triggers a cellular response. So they're not taste receptors per se, because they're not linked to our perceptive pathways the way our taste buds are. However, they are local chemoreceptors in the body, which means that they detect changes in the environment and trigger a signal. We're going to talk a little bit more about kind of the role that our bitter, quote unquote, receptors play in our respiratory health. So just a cool fact about your body, you actually have sweet and bitter receptors throughout your body, and their purpose is to make sure that they're communicating around them and to be on the lookout for their particular kind of material. All right, so this is, if one thing to know about me is I'm a huge Simpsons fan. This is one of my favorite Simpsons jokes. It said, I caught my wife cheating, and he said, "You bit her?" And he said, "Yep, bit him too." I think about this on a daily basis. I'm not even joking. So our bitterness sense comes from what are known as type 2 taste receptors. So those are a distinct class of those GPCRs. One interesting area of research now is whether bitter molecules taste more or less the same. And that's a question now because we may be able to distinguish different classes of bitterness, but it's difficult to tease out because we don't generally experience isolated bitter tastes. For example, you may really love IPAs, but you might find grapefruit to be too bitter. So that suggests that we can detect different types of bitterness, right? Otherwise, if there was just one bitter taste, liking something bitter like an IPA would presumably mean that you liked the bitterness in grapefruit as well. So for something like sweet, we only have 3 genes to detect sweetness. We have at least 25 different types of bitter receptors. So it's very complex. It's much more complex than any of our other taste senses. Each bitter receptor sends a signal along the same bitter channel. So that's why there's a question now of, well, if all bitterness goes to the same channel, then do they all taste the same? Or do they taste different? And if they do taste different as humans, are we able to detect those differences? And that's where that well, I like IPA, but I don't like, broccoli or something. That's where that comes into play, because each of those 25, at least 25 different receptors is sending the signal in the same channel. So this may mean that bitter molecules taste more or less the same. The reason why we have such a complex system for bitterness is that nature tries to poison us in a lot of different ways, and many poisons taste bitter, hence the need for many different types of bitter receptors. And we'll also talk in a second about different geographical differences between people with different types of bitter receptors. But that's again, are very educated guess on why we have so many different receptors for bitterness. Okay, so next we're going to talk about the concept of super tasters and just the concept of taster status in general. So you can see here in this picture, and I should have mentioned at the beginning, there's going to be a lot of tongue pictures in this. So if you're not into looking at pictures of tongues, I'm sorry. But there will be a couple of more. So you can see here in this picture, a super taster, what that means is that it has more fungiform papilla on the tongue than a non-taster. We're going to get to super tasters and non-tasters in just a second. So people can be broken down into three groups of taster status. You can be a non-taster, which means that you're not sensitive to bitterness. You can be a taster, which means you are sensitive to bitterness. Or you can be a super taster, which means that you're extremely sensitive to bitterness. And like I mentioned, super tasters, all that means is you have a higher than normal concentration of taste buds on your tongue, particularly bitter and sour. It doesn't mean, like, if you're a super taster, it doesn't mean that you're inherently better at tasting. And a lot of times it may actually be an Achilles heel, because I've read about how being a super taster is like you see the world in neon, whereas somebody who's a non-taster sees the world in pastels. So a lot of times people who are super tasters will avoid things that are, have strong bitterness or have a lot of strong flavors. So super taster doesn't mean that you are super at tasting. It means that you have a lot more, higher than normal number of taste buds on your tongue. And your taster status can be determined by what's called probe paper. And in just a moment, I'm going to have you all pull out your probe paper and find out what your taster status is. First, let's go through some taster facts. So this is another tongue picture. Sorry, it's blue, and the reason why it's blue is because of using blue dye to stain your tongue and then count or kind of look at the number of fungiform papilla that you can see is a common way also to kind of get an idea for what your taster status is. So non-tasters, if you are a non-taster, that does not mean that you are blind to bitterness. If you find out you're a non-taster, you're going to be like, well, wait a second, I can still taste it. Yes, and we'll talk a little bit more about that in just a second. All of that means is it takes an increased level of bitterness to be present before you can perceive it. If you're a non-taster, you also tend to be insensitive to other flavors, and you have higher threshold for enjoying things like spicy foods, like beer, coffee, other bitter things. So if you're a non-taster, again, it just kind of means like some flavors can be in pastel for you, whereas if you're a super taster, those flavors are going to be neon. Tasters and super tasters are more sensitive to certain bitter compounds, and they may perceive bitterness at a lower threshold than most of the population. Another interesting note about non-tasters is that since they are insensitive to other compounds, they are also less sensitive to compounds like diacetyl. And I bring that up because knowing a person's taster status can be an interesting data point to know for sensory reasons. And, both the sensory programs I set up previously, this was something I did with my panelists, was had them do the probe strips like we're going to do in just a second. So I knew who among my panelists was a non-taster, who was a taster, who was a super taster. And the reason for that being, if you're a non-taster, you're less sensitive to diacetyl. So let's say that you have a non-taster sensory panelist who says, I don't detect diacetyl in this sample. It may mean that the concentration is not high enough for them to detect, and not that diacetyl is absent. So if you're doing some kind of sensory panel, it is worth knowing what your panelists' tasting statuses are, because you may, if you're like, Jen says she's a non-taster, so I'm not so sure, I'm going to go find a taster to tell me about if there's diacetyl in this. It's just a good data point to know. Also a good reason why you should have a lot of people from different areas on your sensory panel. I won't go on my soapbox about that, but just know that I have opinions about proper sensory panel setup. All right, so the next thing Luckily for you, you all happen to have some prop paper strips, so you're going to find out what your status is. So you should have received a baggie with the label that says probably not drugs in your vial box. And inside each baggie, there are 2 strips of prop paper. It's filter paper, so it's very thin and it tends to stick together. Some of you may have ended up with like 10 strips, but just know that there's more than 1 strip in your bag. And for this exercise, you only need 1 strip of the prop paper, so you can either use the second strip as a second opinion if you don't like what you get the first time, or you can use it to find out the taster status of someone else. So what I want you to do is scan this QR code or go to this bit.ly link. You're going to take 1 probe strip from the baggie, and all you do is place the strip on your tongue. And when I say place a strip on your tongue, I just literally mean open your mouth, stick out your tongue a little bit, put the piece of paper on your tongue. It's going to taste one of 3 ways. It's going to taste like paper, it's going to be bitter, or it may be extremely bitter. Prop only tests for one type of bitterness. So I want you to remember that because I have seen people do their prop test and find out that they're a non-taster, and it can be devastating for people. So just remember, we have all of these different types of bitterness that we can detect. So prop only tests for one type. Being a non-taster with prop paper for bitterness means that you are not blind to bitterness. It just means that you need higher levels of some types of bitterness. So if you go to that QR code or the website, it's just super quick. You don't have to put your name on it if you don't want to. If you don't want to fill it out, you don't have to. I like to just give people prop paper randomly, so I know I know who among my friends is a taster and who's a non-taster, because that's just the kind of friend I am. But if you would like to, you can fill that out on the website. And I think I also have it on there, like, don't overthink it. It's when you touch it to your tongue, you're going to know if it tastes like paper, if it's bitter, or if it's super bitter. So while you're completing that, I will tell you a story that seems to happen frequently in the science world. And that is, I promise it is related to our probe paper. PTC, which used to be used instead of prop to test for bitterness sensitivity, was discovered by accident. So in 1930, there were 2 chemists working in a lab at DuPont. They were working on formulas for blue dyes. So one of the chemists was pouring this PTC powder into a container, and he kind of mishandled the bottle, and it sent a puff of the PTC powder into the air. Both chemists accidentally inhaled the powder. One chemist had a very strong reaction and found the powder to be extremely bitter. That was very surprising to the other chemist because he didn't taste anything at all. So once they tried it again a couple of times with just the 2 of them, this theory begins to form that, oh, wait a second, if it's really bitter to you, but I can't taste it at all, right? There's a there. And so they started asking all of their colleagues to taste this powder. And that's how we figured out that there are super tasters, tasters, and non-tasters. Other things that were discovered by accident, by either inhaling something in a lab or forgetting to wash your hands, also includes artificial sweeteners and LSD. So those are, I don't know, I maybe don't wash your hands. That's, we've gotten some cool stuff out of that. Okay, so humans and bitterness. If you are a non-taster, don't despair. Prop paper only tests for one receptor, which is T2R38. Another bitter taste receptor is TAS2R14. This is also a G protein coupled receptor. And this TAS2R14 can actually identify more than 100 distinct bitter tastants. So this T2R28, 38, only detects this chemical group that's called a thiourea group. Bitter thiourea compounds are also found in broccoli and Brussels sprouts. So if you are a taster and or super taster, and you also hate the flavor of broccoli and Brussels sprouts, this is probably why. The ability to taste probe is genetic. So if both of your parents are non-tasters, you will also be a non-taster. If both parents have the super taster gene, you're also going to be a super taster. If one parent is a taster and the other is a non-taster, you're just a regular taster. And interestingly enough, since this is genetic, when these bitterness tests, when we discovered that there were different tasters, different taster status, they wanted to use it for paternity tests because you could see if the one parent was a taster and the other one was not, then you might have a regular taster. But if you didn't, maybe you're both non-tasters and somehow your kid is a taster, that might tell you something. So bitterness can be an acquired taste. And I think most of us know this. I mean, you're here on a webinar about beer. And I'm guessing that the first time you sipped beer or coffee, it probably wasn't an enjoyable experience. So why do we keep ingesting it? When we think about bitter things that we like, bitter chocolate, beer, gin and tonics, coffee, one reason why we may keep coming back and giving bitterness a shot is because with those things, there's a reward associated with it, right? With coffee, you get caffeine. With beer and gin and tonic, you get alcohol. So we're not necessarily becoming acquired to bitterness just because we love bitterness. We've really become acquired to bitterness because we like the other things that come along with it. So, like we just kind of tolerate bitterness until we get to a point where we like it. So babies are extremely sensitive to bitterness. This is, we talked about this just a few minutes ago. It's part of that, like inborn, you're going to have an extremely sensitive sense of bitterness, particularly when you're young, because that's also when you're going to be the most vulnerable to things like infection, to things like poisons. It's not going to take that much to make you very sick or to poison you. So babies are more sensitive to bitterness. As we get older, we get less sensitive to bitterness. We've already talked about this. So a few interesting taster status facts. Tasters are less likely to develop sinus infections than non-tasters. This is because we have bitter receptors in our respiratory passages. So remember when we were talking about the extraoral, with our extraoral sensors that we have, those bitter receptors, those are also in our respiratory passage because the air that we breathe has bacteria in it. And one of the chemicals used by bacteria to communicate actually has a bitter taste. So our bitter receptors in our sinuses and airways detect that bitter, quote unquote, taste and communicate that to our immune system, telling it that they're intruders. So if you're a taster, your body is going to be better at detecting potential infections coming, whereas if you're a non-taster, sorry, you might be more susceptible to sinus infections. Oh no, I went too far. Nope, I didn't go far enough. All right, so forgive the binary here. Previous science research didn't account for what we now know as a spectrum. However, women are more likely to be tasters than men. There's also geographic differences in taster status, which I think, again, makes sense when we look at bitterness from an evolutionary standpoint, because we developed these bitter receptors based on the poisons found in our environments, right? There's different poisons everywhere trying to kill us. And so there are geographic differences. I have a very pseudoscientific theory about one of these in particular, and potentially the creation of beer styles and how certain beer styles grew in certain geographical areas. But I am not a doctor or a researcher, and this is just me throwing out half-baked ideas. But more than half of the Indian population is non-taster. In Northeast Britain, nearly a third of the population are non-tasters. that's kind of where my idea of like, okay, like we've got, IPAs, pale ales, and such coming out of England. Maybe it's because there was a higher proportion of people who aren't as sensitive to bitterness, so they were creating more bitter beers. Again, that's just my theory. Whether that's true or not, who knows? Time will tell. And as much as 95% of the population in some parts of China are tasters. So that's just a really interesting, to be able to look at the world. I would love to give the world paper strips just to see kind of where those concentrations are and if there are any other interesting kind of connections there. All right, so I'm going to stop talking about our sense of bitterness in our mouth. We're going to turn our attention to beer and bitterness. Out of the four main ingredients in beer, malt, hops, water, and yeast, most of the bitterness found in beer is commonly from hops. Malt and water can also contribute bitterness. We'll talk about that in a few minutes. So specifically, the alpha acids found in hops contribute much of the bitterness that we find in beer. These all fall under the umbrella of humulones. So you may see them collectively referred to as such, but there are three types of humulones found in hops. The first is going to be humulone. That can account to 40 to 80% of our hop alpha acids. Cohumulone will be 14 to 50%. Adhumulone will be 5 to 15%. So those are our hop alpha acids. Alpha acids are not bitter or soluble in beer on their own. They need to be isomerized to provide bitterness. So isomerization is a process where a molecule is transformed into another molecule that contains the exact same atoms, known as isomers, but they're arranged differently. So isomerization is both time and temperature dependent. For our temperature, the isomerization is going to occur when our alpha acids are heated in a solution. For our purposes in beer, this occurs during the boil. The longer hop alpha acids are exposed to boiling, the more they are isomerized and the more bitterness is created. So hop alpha acids begin to isomerize around 175 degrees Fahrenheit, about 79 degrees Celsius. And what's important about this is if you've got, let's say you've got your rolling boil, so you're around 212 degrees Fahrenheit, 100 degrees Celsius, your hops will isomerize. The other thing to keep in mind, though, is that if you're adding whirlpool hops, you can also contribute bitterness if you do not cool down your wort below 170 degrees Fahrenheit before adding your whirlpool hops. So if you're not familiar with hop dosing terminology, now that I just said all that, I will give you a quick overview. When we're talking about bittering hops, these are going to be hops with high enough alpha acids to achieve our desired bitterness level. They're also added during the boil, so those alpha acids were isomerized. Your bittering hops, maybe you add them just once, maybe you add them at different intervals. So for example, if we've got a double IPA, we may have hop bittering additions at like 60 minutes, 30 minutes, 15 minutes. You'll still get some flavor and aroma from bittering hops, but not at the same level as you do with aroma/flavor hops. Bittering hops are primarily used for, surprise, bittering. Aroma or flavor hops are used to add aromas and flavors to beer and not to add bitterness. They are typically added toward the very end of the boil, during the whirlpool, during active fermentation, or after fermentation is complete. So as already mentioned, depending on the temperature of the wort, when the aroma/flavor hops are added, there may be some isomerization that happens. And isoalpha acids also don't just contribute bitterness, they help stabilize beer foam and they help inhibit the growth of bacteria. So when our boil is done, when we've isomerized those alpha acids into isoalpha acids, that bitterness is 4 times more bitter than the alpha acids. So that gives you an idea. Regular alpha acids, no big deal. Once they've been isomerized, they're going to be a lot more bitter. So let's move on to something that drives me nuts, and that's the misuse of IBUs, or international bitterness units. IBUs are the internationally agreed upon standard for measuring bitterness in beer. It was created to assist brewers in producing consistent bitterness in their beers. And that's an important point. That's going to be a thread throughout, that it's not necessarily to tell brewers exactly how bitter the beer is. It's a tool so there's consistent bitterness in their beers. That's obviously going to be important if you're making the same beer more than once. You want it to be consistent, right? You want your customers to be able to rely on that. It was never intended to be a marketing tool. As a marketing tool, it is grossly inaccurate. And I promise I won't go too far off on this, but breweries, particularly craft breweries in the early days of craft, putting the IBUs on cans has like destroyed what we know, what we think we know about bitterness versus what the actual purpose behind IBUs is and what the actual perceived bitterness is. So grossly inaccurate communication about the perceived bitterness of beer. So the IBUs, these are going to be a calculation of the quantity of alpha acids in wort or beer multiplied by the fraction 5/7. So the IBU method of measuring bitterness was developed in the 1950s and 60s and reflects the beers of that day. So think back 1950s, 1960s beer. Back then, most breweries did not have access to fresh hops, so some of the hop bitterness actually came from oxidation products in the hops. Since not all of the hop bitterness could be attributed solely to isoalpha acids, measuring isoalpha acids only would not accurately reflect the total bitterness contributed by the hops. So that's where this 5/7 fraction comes into play. It's a correction factor. The 5/7 fraction is based on the assumption that isoalpha acids were responsible for 5/7 of the bitterness of an average beer in the 1960s. The remaining 2/7 of bitterness came from non-isoalpha acid hop material, most of that from aging hops. So cold storage is now the norm for most hops. There's also better hop products. So the utility of the IBU is not as useful a measurement as it once was. It's a lab procedure, not a marketing tool. Along those same lines, as an evaluator, I strongly would love it if when you're evaluating beer, don't try to say what the IBUs are. Don't try to say what the SRM is. You're not a lab instrument, you're a human. We have lab instruments that will tell us things like IBUs, things like SRM. What your role is as an evaluator is to give me the type and the intensity of the bitterness. What is the perceived bitterness? We'll get into that in just a few minutes, but obviously IBUs are not the way to communicate how expectations around the bitterness of a beer. So not only the quality, the overall perception, like I just said, IBUs do not reflect either one of those things. So other limitations of IBUs, and I promise, I'm not hating on IBUs. I'm hating on the people who told people that their beer was 75 IBUs or whatever. Anyway, so the theoretical saturation point of isoalpha acids in beer is about 110 IBUs. Once you do that 5/7 math on that, that's going to give you, I don't know, I'm just going to say like 70 some IBUs after we correct for that oxidative factor. So this will correspond to actual isoalphas if and only if, and this is a quote from Val Peacock. This was in the Stan Hieronymus Hops book. I've got that source at the end for you. But it corresponds to isoalpha acids if and only if the tested beer was made with hops as equally deteriorated as the hops that were used in the IBU calibration. Another limitation of IBUs is that wort IBUs and beer IBUs are two different values, mostly because of that drop in pH during fermentation. that's going to change our IBUs. And the yeast will also absorb a certain amount of that excess IBU, that excess bitterness, those isoalpha acids. And if I remember correctly, I think the stat is like when you pull yeast, spent yeast, and examine it, it is usually about 3 or 4 times more bitter than like what the beer is, and that's because those isoalpha acids are flocculating out during fermentation. So even if you have, let's say, as a home brewer, I use Beersmith, it will give me an approximation of IBUs. What that's giving me is my wort IBU, not my beer IBUs. The other thing is the that change, that drop in pH during fermentation, that's going to lower the amount of IBUs in a beer. That's still not completely understood why that happens. So bitterness units and the amount of isoalpha acids are equivalent only in the range of 15 to 30 IBUs, and that is only if the hops are fresh. So those IBUs are not a one-for-one with our isoalpha acids. And when we're talking about, we have better cold storage, people have more education on how better to handle hops. We're getting hops a lot fresher than used to be available. How useful is this international bitterness unit now? Is it accurately reflected in what today's current, brewing world looks like? But again, the purpose of it was to help brewers make consistent product, not necessarily to help brewers precisely measure the amount of bitterness in their beers. So one of the biggest, or I would say the biggest, component to the bitterness in beer is going to be our hop utilization. So like I mentioned, our IBUs function mainly as a quality control measurement for brewers. We can get some information about the intensity of a beer's bitterness, but we will talk about some other factors in just a moment that also affect perceived bitterness. Estimating IBUs of a particular beer can give brewers a number to aim for to ensure consistency in the brand and to evaluate whether a specific batch is considered true to style. So calculating hop utilization is the first step in estimating our IBUs. For most breweries without an in-house high-pressure liquid chromatography, HPLC, hop utilization is still going to be an estimate. So there are several variables in determining our hop utilization. The first one is going to be the form of hops that we're using. Are we using whole cones? Are we using pellets? Are we using extracts? If we're using pellets, are we using, like T45? So whatever your hop form is, that's going to be important, in particular because things like hop pellets are about 15% more efficient than cones. So our boil time, we've already discussed how the boil time affects our hop utilization, but one more note is that isoalpha acids will break down into undesirable components after 90 minutes. And that can definitely come up if, for whatever reason, you need to extend your boil. Keep that in mind that after 90 minutes, those hop isoalpha acids that we have in there to provide us bitterness can actually end up giving us unwanted flavors. Boil temperature, water boils at a lower temperature and higher altitudes, which can lower utilization. There was an experiment conducted, I think, at OSU in the Shellhammer lab where they boiled like low alpha acid hops for a very, very long time at a low temperature and like eventually got some bitterness. But generally speaking, 175 degrees Fahrenheit or above, that boil temperature is going to matter in terms of our utilization. Kettle geometry, larger kettles are more efficient than smaller kettles, than things like carboys. Our wort gravity, our utilization is going to decrease as wort gravity increases quickly. What that means is more or less when our alcohol level is going up, our hop utilization is going to go down. pH and the mineral content of water, your efficiency increases with pH, but it can also be detrimental to trub formation, protein composition, and yeast nutrition. So we run into this a lot, right, when we're learning about beer. This is one of many examples where a brewer determines where to make compromises in the brewing process based on the ultimate goal. So yeah, we do have the technology, or we do have the science and the know-how to make our hot liquor, a little higher in pH if we want really good hop utilization, but doing that is going to have an effect on all those things: trub formation, protein composition, and yeast nutrition. So is it really worth it? The humulone composition, higher cohumulone is more efficient. There is also, you may see sometimes there's anecdotes out there that brewers think that cohumulone gives kind of a rougher, harsher bitterness, and that humulone itself is preferable. But when like actual experiments, like blind panels are done, nobody really sees a difference between the bitterness that comes from high cohumulone and the bitterness that comes from humulone. That doesn't necessarily mean that it's not there. It's just that has just only been an anecdote and not an actual like proven fact. So let's talk about how we can estimate our IBUs. For purposes of this webinar, we're going to make some assumptions about the variables in our formula. We are going to assume that we do not have access to exact measurements such as HPLC analysis, which many breweries do not have. So this is our IBU calculation. We're going to take the quantity, which is going to be the amount that we're using, multiply it by our alpha acid percent. That's going to be expressed as a whole number, not a percentage. The same thing with utilization, whole number, not percentage. That 0.7749 is a correction conversion from the metric system to whatever this is. And we're going to divide that by our beer volume. So in terms of quantity, that's just going to be like, how many ounces are you using? Our alpha acid, that is typically expressed as a number on your hop package. However, we don't know necessarily where that alpha acid number comes from. It might be the alpha acid percentage at the time of harvest. It might be the alpha acid percentage at the time of processing or the time of packaging. Your hop manufacturer should be able to give you that information if you ask them for it. I'm not sure if they would be able to do that like with specific lots or anything, but anyway, your alpha acid percentage, you're going to find that on your hop packaging. Usually when you look up hop varieties, they'll also tell you kind of what the average is going to be for that, or the range for alpha acids for that hop variety. Utilization is tough to do. It is going to be the amount of alpha acids added to the kettle versus the amount of isoalpha acids that we get from the wort. We do have a hop utilization percent formula. That's going to be your isoalpha acids in wort times 100 divided by the alpha acids added to wort. Again, the only way you're going to know what those isoalpha acids are is the actual amount is if you have something like an HPLC, which is going to tell you that amount. That hop utilization is or particularly those isoalpha acids. Again, that is a laboratory measurement. And that's also something that a lot of brewers, particularly, well, probably only craft brewers, don't have access to, sophisticated lab equipment like that. So generally speaking, it's safe to assume like a 25 to 30% utilization for when we're trying to figure out this, how many IBUs about we might have in our wort. Typically when I'm doing the conversion for this, or doing this IBU calculation, I'll just assume like if I'm doing it on my homebrew system, I assume like 20%. If you've got a professional system, obviously it's going to be more efficient. You're going to get a little bit more utilization out of it. But just know that in the real world, most of this is just the utilization is going to be assumed. And our beer volume, typically when you're doing this calculation, the standard is to just figure it out by 10 gallons and then, scale that up depending on the size of your system. So let's do a quick example. For this example, we want to estimate how many IBUs we'll get from using 2 ounces of Chinook pellet hops as a 60-minute bittering addition in a 10-gallon batch of beer. So to start, our hop quantity is going to be 2 ounces. That's literally just a number I made up. It's not significant or anything. I was just trying to think like, okay, if I want to end up with a kind of good IBU, how many ounces would I want of something like Chinook? So we've got here the information provided by our supplier. The alpha acids in Chinook are about 12 to 14%, somewhere in there. So we're just going to split it and assume our alpha acids are 13%. For utilization, I'm doing this at a professional brewery. I have more efficient equipment. So that's going to be, we'll just say 30%. So we're going to plug all this in, beep, beep, boop, 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 boop. And we can see that using 2 ounces of Chinook hop pellets with 13% alpha acid in a 10-gallon batch of beer will give us approximately 58 IBUs. Keep in mind, however, that this is only an estimate, and this is only an estimate of our wort IBUs. It's not going to tell us anything about our final beer IBUs. The other thing to keep in mind about this is if we're using 1 bittering hop at 1 dosing time. So if I'm doing, let's say, even if I'm doing Chinook, all Chinook, but I'm doing a 60-minute, a 30-minute, and a 15-minute, I will need to calculate the IBUs for each one of those additions. So let's talk about what really matters, and that's perceived bitterness. As I mentioned earlier, there are other factors at play in the perceived bitterness of a beer. From a sensory and consumer standpoint, this is much more important than putting what is probably definitely an inaccurate IBU number on a beer label. So some other factors that will affect our perceived bitterness. Perceived bitterness, again, this is what we perceive. What I'm looking for when I'm talking about perceived bitterness is the intensity and kind of the ratio of bitterness within the entire beer. So malt obviously adds sweetness. Roasted malts can also add bitterness. Water, depending on how you treat your water, if you've got something that's high calcium sulfate, that gives that quote unquote crisp bitterness. I despise crisp as a descriptor because what does that even mean? But it will give it a crisp bitterness. Something that's high in calcium carbonate, it can be very coarse bitterness in high levels. So, your sulfate to carbonate ratio, that comes into play, particularly when we're talking about like hazy IPAs and, wanting kind of a softer, rounder mouthfeel. I also do not like those as descriptors. But if your calcium carbonate gets too high, it will cause a gross bitterness in high levels. Actually, if you have too much calcium sulfate, it would also add just a bad bitterness. Polyphenols, we get polyphenols from malt husks and from hops. Those will also affect the bitterness in the beer. Our beer temperature. Lower temperatures suppress bitterness. So if you have a super cold beer, it's not going to taste as bitter as it will as it starts to warm up. Our bitterness ratio, this is calculated by dividing estimated IBUs by original gravity. You can see here on, this is a chart from Beersmith, and I forgot who he had credited for this, but you can see our bitterness ratio and kind of how that changes as your ratio gets greater or gets lower. Like I mentioned, our alcohol content, higher alcohol beers require higher levels of bitterness for a balanced flavor profile. Recall also, though, that as your ABV goes up or as your gravity goes up, your hop utilization is going to go down. So you need to not only account for needing a higher level of bitterness in a higher ABV beer, but you also need to account for the fact that that higher ABV means that your utilization isn't going to be as good for your bittering hops. Attenuation. High attenuation means higher perceived bitterness. Why is that? Because if something is very highly attenuated, we don't have a lot of residual sugars left in there, so we're going to have higher perceived bitterness. Whereas if we have a low attenuating yeast strain, we're going to have lower perceived bitterness because more yeast, more sugars are going to remain in that final beer, and you're going to have those residual sugars in the beer, which is going to lower your perceived bitterness. And this one's really interesting. So hop, oils, and aroma, these can enhance the perception of bitterness. However, this is actually just the note on hop, oils, and aromas enhancing the perception of bitterness is actually a sensory bias, and that's known as a logical error. So a logical error occurs when we build an association in our mind between two distinct characteristics. In this case, hop aroma and flavor and bitterness. We tend to blend those two together. So if we have, a lot of hop aroma and hop oils that we're detecting, that's we are, excuse me, pardon me, we can learn to blend that into higher hop aroma is going to be higher perceived bitterness. That may not necessarily be the case. So one way to try to mitigate this logical error is doing individual attribute training. So that's going to be, where you're looking at bitterness separate from aroma. One way you could do this is, let's say you have a low hop aroma beer, you can get some quinine, which is going to increase the bitterness, the perceived bitterness, and, kind of train yourself that hop aroma is not correlated to hop bitterness. They're two separate things. So that is, I believe, yes, the end of our slides. And what we're going to do next is go into our second blind smelling exercise. So what I would like you all to do, your vials, you should still have them in front of you. Just kind of move them around, switch things up so you're not necessarily tasting in the exact same or smelling in the exact same order. This time is going to be a little bit quicker. We're only going to take 3 minutes to do this blind smelling exercise. We're going to use that exact same form that you used before. I will stop talking and I will be back in 3 minutes. Also, make sure you hit submit before you get finished. Okay, you have about a minute left. All right, so that has been 3 minutes. Go ahead and finish up. If you haven't finished already, make sure you hit submit on that. And very shortly, we will go on to our flavor reveal. So as a reminder, because we're going to wrap up here in a couple of minutes, if you have any questions, you can put them in the chat box and we can answer them. Of course, if you would prefer to just email me directly with your questions, you're more than welcome to do that, or if you just think of something later. So with our flavor reveal, what we're going to do, and I started to do this last time and then was flustered by not having any audio. So what I would like for you to do is when we go through each one, I'm going to tell you the number, I'm going to give you a second to find the vial with that number, and then I'm going to tell you what the aroma is. Now, when we do this, the other thing I want you to do after, sorry, I'm trying to talk and find my vials at the same time. So when I tell you what the aroma is, I want you to take a couple seconds, smell it, and just say what it is. So let's say that this is yuzu. This is not yuzu, but I'll smell it, say out loud yuzu. The reason for that being, if we speak something out loud, it makes a stronger memory connection for us. So for each one, I will tell you the number, give you a second to get the number while repeating that flavor out loud. If possible, if you're somewhere where it'd be super weird for you to be like chanting yuzu, you don't have to do it. But know that when you say things out loud, it does strengthen that memory connection for you. So let's go. Our first one is going to be sample 752. Sample 752 is grapefruit. Okay, our next one is 574. 574 is lemon. Next, we have 308. 308 is lime. And last, we have 355. 355 is mandarin orange. So hopefully you all feel pretty good about, sorry, I've got citrus one all over my nose now. But I hope you all feel good about the citrus flavors and how you did. If not, remember, this is progress, not perfection. And what we're doing, just going into this really completely blind, is super tough. We're doing some really advanced kind of training here. So don't be frustrated if you didn't get any or you just got one or two. That is no big deal. The purpose of this, first of all, is to get you used to doing things like blind aromas and trying to deduce what they are. Now that you know what they are, you can go back, mix them up, smell them again. And the more you practice this, the easier it's going to get for you to be able to identify these. As always, here are all of the sources that I used today to prepare. All the information in this presentation came from one of these sources. And let me see. I think that's really it. I don't see any questions. You all have like 30 more seconds to ask any questions if you have them. If not, then we will go ahead and wrap up. So our next webinar is going to be Citrus 2, and that's going to be on June 26th. I can see from the banner I made myself at 8 p.m. So you can find all of the future webinars on the website at Aroma Explore the Wheel under the Ginfluence.be or Aroma Explore the Wheel. You can see all of those dates and everything upcoming. I will email you all the link to this video as well as a copy of these slides likely tomorrow. And if you have any questions in the meantime, as always, you are completely welcome to reach out to me. And I'll do the best I can. I can usually do a pretty good job with questions. And in the follow-up email that you'll get, there will also be a link for you to leave me feedback if you would like. It's very important to me that you find value in each one of these webinars. So if there's something that you think would make the webinar more helpful or more valuable, let me know. If there's something that, again, I forget that not everybody lives inside my brain. So if there's something that you think going through could use a little bit more explanation, let me know that too. Seriously, as long as you're not mean, you're not going to hurt my feelings about it. And if you also are so inclined, you can write a brief testimonial for me. I always appreciate when people do that. So thank you so much, everyone, for joining me this month for Aroma Explore the Wheel, Citrus 1. I hope you enjoyed it, and I hope you all have a great rest of your week. Bye.